So triple negative breast cancer accounts for about 20% of breast cancer. It's probably the most malignant, and it affects younger women. And it's described as what it isn't. That is, it is not ER positive, it's not PR positive, and it's not HER2 positive. But what we and others are now finding that within this subgroup is actually heterogeneity. But what I'm going to describe is how that heterogeneity can be abstracted into very characteristic genomic configurations that has clinical meaning as well. So it began with analysis of whole genome sequencing. By the way, not exome sequencing, but whole genome. And we were specifically interested in structural mutations in breast cancer. And what we were struck by was a number of breast cancers had a unique configuration that was multiple single tandem duplications. These are segments of DNA that are duplicated in a head-to-tail fashion, but only duplicated once. Okay? But that happens all throughout the genome, your genome, other cancer genomes. But one thing we found very unusual was that this occurred very frequently, in the hundreds. And they were spread out throughout the entire genome as opposed to concentrated in a hypermutable area. This was so unique because you see this and it becomes obvious that something is wrong, that we decided to do it in a computational and mathematical manner, because otherwise it's like asking, is this picture like Matisse or like Rembrandt, you know? Unless you can deconvolute it into a mathematical principle, it's hard to look at it across this complexity across all tumors. So we made a very simple measure of the number of tandem duplications and how evenly distributed they are in a genome. And the thing that surprised us is that it was not a Gaussian distribution. It was actually a subgroup of cancers that had a very high mutational rate based on the single tandem duplications across the entire genome. When we looked at it carefully, now we extended that study to about 2,700 tumors that have been sequenced. We found that they are uniquely clustered in certain tumor types. In breast cancer, only triple negative breast cancer, not the other types, had this. And triple negative breast cancer was 50% had this kind of many tandem duplications. 50% of uterine, 50% of ovarians had this. Equally important was that prostate, colon, leukemias, thyroid cancers, and brain tumors were completely devoid of this tandem duplicator phenotype. Let me make it clear, tandem duplications occur, but in the frequency and the distribution that it has, it's a very unique subset. Then we started to dig down deep in terms of the characteristics and found uniquely that these tandem duplications can be further segregated into the size of the tandem duplicon, what we call span size. Instead of saying, oh, it could be 1, 10, 15, 20, 100, 1,000 kilobases, we found that they were clustered in individual tumors. So some tumors that are tandem duplicator phenotype tumors, TDP, that had many of these, right, clustered around span sizes of 10 kilobases. That the hundreds that they have are all around 10 kilobases, plus or minus 5 and 10, okay? And there was another class that clustered around 230 kilobases. And then the third class, which is rare, that clustered around 1.7 megabases. Okay. That obviously told us there is unique biology in terms of its restriction into tumor types, and unique biology in terms of the clustering of the tandem duplicons. So we began to look at it carefully and found that of the TDP tumors coming out of 15% of all tumors that we looked at, of the 2,700 tumors, had a TDP, of which it was enriched in triple negative breast cancer, ovarian, and uterine. When we looked at those, we found that individual tumors can have either this 10 kb variety the 230, the 1.7, but they also frequently mixed, but they mixed only in binary fashion. So the 10 kb and the 230 kb occurred within a single tumor. The 230 with a 1.7 megabase occurred in one tumor. But these combinations, either the unique ones or combination, accounted for almost 100% of all the tandem duplicator phenotype tumors. Now, was very important for us to find out by using this approach to separate out the different TDP types, that we were able to then to look at the originating mutations that generate these tandem duplicator phenotypes. So we found that the short tandem duplicator phenotype, which we call group 1, were exclusively BRCA1 and mutant and P53 mutant. Now, the important part of this is that it is not just a germline carrier. In fact, about 50% over 50% of the type 1 TDP, the one with the 10 kilobase span, were somatic mutations or promoter methylation of the BRCA1. And for the 230 kilobase variety, that had predominantly cyclin E aberrations, inducing augmentation of cyclin E expression, but always with a P53 mutation as well. And then in ovarians, the CDK12 mutations was also a cause of the large span DNA. So we actually generated the hypothesis that specific mutations generated these chromosomal instability configuration. Now, we confirmed this by looking at mouse models. So mouse models that have knockout of P53 get breast cancers. Knockout P53 and BRCA1 get breast cancer. BRCA2 and P53 get breast cancers. And what we found was, of the mammary tumors of these mice, only the ones with P53 and BRCA1, not P53 alone, not P53 and BRCA2, generate this tandem duplicator phenotype of exactly the same configuration that we saw in the human. In a Nature publication that just came out, we worked with our colleagues at Beth Israel Deaconess Medical Center, where they used a in vitro indicator system that they can score tandem duplication formation, and found that in the absence of BRCA1 and not BRCA2, in conjunction with fork stalling, replication fork stalling, and not double-strand breaks, you induce tandem duplications of the same size. So we, in a very short order, identified the mechanism for this, surrounded by BRCA1 deficiency and P53 deficiency, okay, causing a clinical syndrome. It's a carcinogenic process, but causing a clinical syndrome that's relevant in the human. A very important part of this is that we found, using patient-derived xenografts and breast cancer cell lines, triple-negative breast cancer cell lines, that the TDP score is correlated with response to cisplatin chemotherapy. And this is actually very important, because triple-negative breast cancer, unlike all other breast cancer types, is responsive to platinum. And the induction of pathological complete remission is the best indicator for long-term survival. And the big problem is, how do you know pathological complete remissions only occur in between 30 and 50% of patients? And the 50% is when they use platinum and taxane up front. Now we think we have a tool to be able to segregate out those who respond to this therapy versus those who won't. And this allows us, in the first instance, to personalize therapy within the triple-negative subset. So, we're in a process of moving this into the clinic to get that validated. We're very excited about those possibilities. From this is actually very important. What are the downstream consequences of these hundreds of tandem duplications? Well, we studied that. Fascinating is that that probably is the system's induction of a cancer. No single TDP tumor looks alike because these tandem duplicons aren't in exactly the same places, okay? But what we found is that there's a tendency for these tandem duplications to augment the expression of oncogenes and kill the expression of tumor suppressors, but not one, multiple ones, all right? Now, the challenge with that is that some of the oncogenes and tumor suppressors that they augment and otherwise would not be detected by classical diagnostic approaches. Let me tell you why. Normally you look for HER2 amplification, okay? The tools we use actually screen for amplifications at the 5 and 6 fold. That's the threshold. We're talking about a single tandem duplication, okay? There's increasing data to suggest that in the absence of amplification, that there's these HER2-like triple-negative breast cancers that may be responsive to lapatinib. Now, we need to test this, but this could be exactly the reason why some individuals respond in the triple negatives. Now, the other aspect of it is that, for example, P10 is mutated and RB is mutated, but these mutations are not scored by exome sequencing because they are not point mutations. They are actually tandem duplications that interrupt the appropriate transcripts. So what this tells us is that if we're going to look at triple-negative breast cancers, we're probably going to need to have a different set of tools to address the mutational profile, because they're not point mutations, they're actually structural mutations, which are more difficult to discern using the standard approaches that we have at this time. I think one of the key issues here is it's an issue of how you look at the data. The unique aspect of this is that we're not looking at individual genes that are mutated. We're actually looking at a chromosomal configuration. And that just is an indication of the complexities that we'll have to grapple with when we analyze cancer genomes.